So I'm going to give you a little overview of Climb, and I'm going to hand over to Nick. Nick's going to talk to you a bit about science, and then I'll talk to you about where we've taken some of the things we've learned from doing Climb and how we think they can apply to Birmingham. So what's Climb's impact on us taking research forward in Birmingham? So Climb was MRC funded. I think it was an £8.5 million grant across 4 universities. A few people moving around, so it's including Bath as well now. And it's to support microbial bioinformaticians. So anyone in the UK community who's working in microbial bioinformatics can get a VM on Climb. And really here at Birmingham, we built a lot of the pilot service, which went on to define how Climb looks at other sites as well. So the hardware is located across 4 universities. We're all connected by the Janet interconnect in the middle. And there's a web application called Bryn, which is written by Nick and some of Nick's team. And basically, you can come along and you can say, I want a virtual machine in Climb, and it will create it in one of the regions that we've got in Climb. And this is what a typical site looks like from a sort of architectural view. We have a whole bunch of compute nodes down at the bottom. Those are the things that run all of your VMs. They're up to 3 terabytes of RAM, some of those systems in Climb. And each of our hypervisors has got 512 gig of RAM. And we've got up to 16 sockets in some of the boxes. And then there's controller nodes to support it and some very fast interconnect from Mellanox. And we've also got Brocade fabric networks in there as well. I'm going to hand over to Nick to talk about a bit of science.
All right. Okay, thanks very much. So I want to present how we've used Climb so far, but I guess really what I want to talk about is how Climb will be useful in the future, and very much relates to the previous talk in terms of the opportunities having a large cloud infrastructure federated and usable by all manner of different researchers, the opportunities that will give us in terms of understanding biology, practicing medicine. So just some context, I'm really going to be talking about a particular type of data set that we're focused on in the Climb project and in my group, which is genome sequence information. And the history of genome sequencing has distinct parallels to the history of computer hardware and computer software development. It's accelerated. It's all happened in a shorter time period than that of computing. Just 20 years ago, the idea of genome sequencing was a new one, and the very first bacterial genome sequence was finished about 21 years ago. To do that kind of work required very large teams working in genome centers. This is Tiger in Rockville, one of the genome centers that was involved in the original race to the human genome. And on the right is what this kind of factory approach for genome sequencing would look like. Tens or hundreds of automated DNA sequencers working on a chemistry that was invented by Fred Sanger in the 70s. And this is very much analogous to the kind of mainframe model of computing. So you've got a whole bunch of computers and kit working together to generate what would now be considered a relatively pitiful amount of information. At the time would be considered exceptionally exciting. And in terms of economics, that would mean that a human genome, or the first human genome, was estimated to have cost something of the order of $2.7 billion, and that was considered a good deal at the time because they'd budgeted $3 billion to bring it in. And so it took about 13 years to complete, faster than anticipated. And the proof of principle of the human genome was done on bacteria, which is mainly what we're going to talk on microbes. We'll talk about that in my talk. And a microbial genome cost at least $1 million, and take at least a few years to generate. The first one in 1995. If we fast forward through the last 5 to 10 years, we've seen just a rapid reduction in costs of genome sequencing and a rapid increase in output. And there's this quite famous curve now on the right here which, if I presented this at a biology conference, it's so overused now, people will throw stuff at me. I might just about get away with it here with a more diverse audience. But basically what this curve is showing us is the cost, log scale on the right, the cost of sequencing has fallen exponentially and very rapidly since around 2007, 2008. And often what people say is, well, Moore's law talks about the doubling of transistor capacity every 18 months. The sequencing output doubles approximately every 6 months since the introduction of new sequencing technologies, so-called next-generation sequencing technologies, an example of which is on the left from Loman. And this means that now microbial genomes can be done for about $50 in a matter of hours to days, and human genomes quite recently hit that magical $1,000 genome threshold that people always talk about and can be done in days. So that's the background, and if you imagine this is like the IBM PC, a kind of desktop sequencer before we had the mainframe model, well, things have moved forward even since the last couple of years. So now we're in a position, and this is where we're interested in using CLIMB, and this is the subject of my research. We're in a position where we can start to realistically do sequencing in real time for outbreaks or for epidemics or for an unusual, unexplained diagnosis of an infection in a hospital. It suddenly becomes a realistic prospect because it's fast enough and it's cheap enough to kind of threaten or disrupt the kind of conventional microbiological paradigm in bacteriology, that would be culture and sensitivity. And now there are numerous examples, mainly in retrospect or relatively slowly working out either the origins of a particular outbreak. So, for example, we worked on the origins and the evolution of the large outbreak of E. coli that affected a few thousand people in Germany and Europe in 2011, and more recent, and also, for example, the outbreak of cholera in Haiti, which is ongoing. Genome sequencing was used for the first time to determine the source of that outbreak, and in fact that was tracked to UN peacekeeping force from Nepal by comparison of genome sequences. There's a whole library of genome sequences from all over the world, and you can use genome sequences in this way. And we know now that genomes evolve in a reasonably predictable way, and understanding that evolution and visualizing that evolution can give us the ability to perhaps influence the way that we do infection control in a hospital or public health epidemiology on a larger scale. And that's before we think about biology, because biology is another, it's a very, very rich source of biological information, this genome data, that could be used to understand fundamental questions about evolution and about how pathogens work, and also to monitor the responses to our human interventions, like giving antibiotics or bringing a new vaccine. So we know that genome sequencing is very useful. We've got good proof of concept, but in the case of the Ebola outbreak, which I'm going to talk about briefly, and the case of the Ebola epidemic from 2014 to this year, something was very notable. So despite the wide availability of genome sequencing capability worldwide and these desktop instruments that I mentioned, in fact, during this huge epidemic, the largest in history, very, very few genome sequences were made available throughout the outbreak. So on the top left here, we have genome sequences, and on the bottom, we have the epidemic curve of Ebola in 2014, so a peak of around 7,000 cases in October. And there's really mirrored by a very small number of genome sequences, and that's perhaps for extremely good reasons. So there are perhaps much more pressing things to do in Ebola epidemic than doing genome sequencing. For sure, that's true. But that's not through a lack of desire to get genome sequences. We know that genome sequences are useful for understanding pathogen evolution, We know that they can be useful in helping give information to epidemiologists. We know that we can understand interesting biology, and people were clamoring, researchers and doctors were clamoring for this sequence information that was not available. And part of the reason it was not available was because it's logistically extremely difficult to do sequencing in West Africa with conventional technologies. And if you want to do sequencing in genome sensors, in established genome sensors, you have to ship this samples out to these labs, and the logistical and the ethical issues are quite profound. So our idea in this case, and this is something that we started to think about in 2014 and really managed to establish in 2015. This is a kind of a very proud moment for me as a geek, where it was featured by Bill Gates. Obviously attracted his attention of actually doing this genome sequencing with a novel portable genome sequencing instrument, in this case called the Oxford Nanopore MinION, trying to actually take sequencing out into the field and doing sequencing in real time. And so this is a kind of interesting story which I don't have time to go into a huge amount of detail on, but we managed to, this work done with Josh Quick in my lab, we managed to get this instrument packed up and ready to go to do genome sequencing in collaboration with Public Health England and the European Mobile Laboratories, who had a set of diagnostic laboratories set up in Guinea and Sierra Leone. We were able to take this instrument out to Africa to do genome sequencing. And the idea was, could we generate genome sequences fast enough and quickly enough, analyze them, and use that information to help with the public health response and to understand better Ebola evolution? So Josh Quick, pictured on the left, with Mars Carroll on the right, Dr. Masuma, one of the doctors involved in the Ebola vaccine trial here, just 2 days after arriving in Guinea, actually managing to generate genome sequences from Ebola on this extremely portable setup. So you can see it's just a laptop and this USB stick. Very, very portable system, doesn't require an air-conditioned lab, doesn't require continuous power because it's powered off the laptop. And really a very amenable instrument for doing this kind of work in field conditions. So I haven't talked about CLIME at all yet, but CLIME played an important role in this project. This is a completely new style of instrument. It was only released in 2014, and it had a very completely different operation to traditional sequencers, a single molecule sequencer. And that means that the data that comes out of it is very, very different. So this plot is just to illustrate the kind of noisy signal information that comes off these instruments that we have to be able to translate into bases in order to generate accurate genome sequences. And we did this in collaboration. So most of my work's done in collaboration with many different people, and this was a very successful collaboration with a scientist in Ontario called Jared Simpson. And CLIME played an extremely important role here because we were collaborating, and what the nice thing about doing a collaboration with someone in North America is quite efficient because you do some work, and then in the afternoon you can hand over that work to them, and they work on it, and you wake up and there's new stuff to play with. And we were able to collaborate successfully because we were able to share logins to CLIME servers that we were able to provision according to the needs of the project. So we started off writing code and finding out, as is the case with much academic software, it's rather inefficiently written, that uses a lot of memory, maybe uses a lot of CPU, and it means that with our CLIME infrastructure we have this kind of elasticity. So if we've decided that we need more resources, we can just shut that down, start it up, and so we ended up using very high memory servers with a terabyte of RAM, very high numbers of compute. CPUs, so 60 CPUs. Through iterating, we were able to develop an algorithm that was published that made us able to get very good quality information out of this quite noisy data. So there's a kind of a trivial example of how CLIME could be used, but little things like the data sets could get larger and larger, and we were at some point training a model on a 10 terabyte data set. And traditionally, if your server had 5 terabytes of disk and you run out of space, you'd have to wait to go down and get new hard drives installed, and your work would be paused until then. With this, we can just create a new volume and keep working. It's extremely helpful for just moving the process of bioinformatics along. So in the end, the larger sort of idea about how CLIME and cloud computing will help us with this kind of work is thinking about the large-scale integration of these data sets. And so we were doing our sequencing effort. We sequenced about 170 Ebola genomes in Guinea throughout this epidemic, and other groups were also doing sequencing, both in the US and the UK, worldwide, and also in Sierra Leone. And we were able to start putting these data sets together and try and understand how Ebola's evolved. And so this phylogeny is just a way of illustrating how Ebola has evolved from the original case down in the bottom left and generated huge amounts of diversity through random mutations. The Ebola virus genome evolves at about one difference every 2 weeks. And over the period of that epidemic, the viral diversity increases, and we end up with a whole set of sublineages or clades or groups there. And you can see that those clades and groups are color-coded according to where in West Africa they came from, and you can hopefully see there's evidence of clustering. Different countries tend to have different colors. Kind of basic stuff. But we can start to use this information for infection control and for epidemiology. So, for example, with the boxes are sequences that we generated in Guinea in 2015, and we can give this information to an epidemiologist, and we can see very clearly that if a case from a village falls into the lineage at the top, and then we have another case around the same time, and the epidemiologist wants to know whether it's possible that the cases are linked. If they fall into distinct groups, we can say very clearly that they're not linked. And we can start using that kind of information to inform our infection control practices. And the point about this is the point about data sharing that was well illustrated in the last talk, is that we really, if we have our data in silos, and silos is the default method for scientists, and their data sets are often very protective of them. If we look at our data sets in silos and we don't share them, we don't integrate them, we can miss things. So it's just a very kind of trivial example, if you like, but on the left is the sequencing that we did in Guinea. On the right is the sequencing around the same time done by a Cambridge group in Sierra Leone. And if you only look at your own data sets and you can't share and compare with other people, then you'll miss things. And so if you're eagle-eyed, what you can see is when we overlay those sequences from the same time period, there are examples. For example, down at the bottom here, we can see some purple dots interspersed with the blue dots. And these are very clear examples of cross-border transmissions between Guinea and Sierra Leone. We can actually use that information and tell the WHO that this is going on, and they can use that to inform border control policy. And in fact, that's what happened. We were able to use that information to feed back and to say, actually, you've got more cross-border transmission of Ebola going on than perhaps you anticipated. So a genuine real-world output, but it can't happen unless there is some mechanism for sharing and comparing information. And that's really the opportunity we see CLIME giving us. So just to put that into more context, this is the most well-sampled epidemic of all time in terms of the number of cases genome sequenced, about 1,600 now, about 5% of the total number of cases. Because this data is annotated with the place it came from and when we collected the information, we can start to, that's not going well, we can start to do this kind of phylogeographic reconstruction. It's not supposed to do that. It's kind of exciting. But what this is showing us is, by collecting a broad sampling of the outbreak, we can show in quite exquisite detail how Ebola's moved around West Africa, from the forested region in the center into urban areas, and then local spread around urban areas, and then occasionally pinging off into far-flung prefectures, causing mini outbreaks or flare-ups. And this is what we want to do for everything, you know, all the time, in real time, whether this is in a hospital, an outbreak in a hospital, tuberculosis in Africa or across the world, antibiotic resistance, the movement of genes. We want this kind of level of information, this dense sampling, and this level of resolution that we can get from genome sequencing. We want this to be standard practice, both for understanding biology and for understanding how infections spread and controlling them. So genome sequencing now looks different to how it did even a year or two ago. This is a genome center in Nongo that we set up. We've actually set up 4 genome centers, if you like, in Guinea now. One of them continues and is ready in case of any future flare-ups of Ebola or other hemorrhagic diseases. We've moved this into bus format, and we've gone around Northeast Brazil recently, Josh and I and a team of collaborators in Brazil, sampling Zika, sampling mosquitoes, and all this information gets analyzed on Climb, gets put into a real-time pipeline that's running all the time, crunching the data and generating phylogenetic analysis and being uploaded to various different databases. So I don't have time to talk much about Zika, but if you're interested in that project, it's all documented on our website, Zebra Project. And you know, this is where we can take this concept out of this world. Quite recently, there was an example of this MinION instrument that we're very excited about being used on the International Space Station, sequencing DNA in low Earth orbit. You know, although this is quite kind of a fantastical idea, you know, we've looked at this screenshot in quite a lot of detail. Just to check that this did actually happen, and we're pretty convinced that this is a genuine sequencing run which happened at the time they said it did. So it's not like the moon landings or anything, it's actually real. You know, NASA think that this stuff is going to go out to Mars and, you know, survey for alien life. I don't know whether it will or not, but the point is if we can do this sort of genome sequencing like this, we can do it anywhere and everywhere, and we will do it everywhere. It will be part of undergraduate education, maybe in schools. You know, genome data sets are perhaps they don't quite compete with the physicists just yet, but they certainly will do soon. The rate of data acquisition is huge and getting faster. And we know that there's this kind of tension between the traditional models of doing things in a data center or in a genome center and doing things in a democratic way. But what's clear is we need better methods, new methods for integrating large-scale data sets, and we think that Climb is really going to help us with that. And the other point is that, just a more trivial point, you know, to do this kind of work requires access to cutting-edge software, usually written by academics, i.e. not very good, difficult to install, hard to use, and we need the flexibility that cloud gives us to make this available to people. And just that last point, really, on availability, this week we've been training a new cohort of postdoctoral of graduate students, part of the Wellcome Trust antibiotic resistance doctoral training program. And all of these new students have a group that they share on Climb. They have this self-service interface to start and stop servers. And for the first time, the new PhD students will get their own server with all software and pipelines that they might want to use pre-installed. And they've got that for the duration of their course. And it will be interesting to see what they can do with that. And that's coupled with quite a bustling forum with tutorials and discussion as part of the Climb project. So I'm just going to put that there as an overview of what Climb offers people. Quite a lot of stuff. And I'll pass back to Simon because I think I've spoken for too long. So thank you very much.
Thanks, Nick. So Nick just put a slide up about, you know, what cause, how much memory is in Climb. And we had a discussion at lunchtime is, you know, are we actually going to tell you what's running in bare cloud? And kind of part of my view is it's a cloud and you shouldn't really worry about what's in there. So we've not talked about it being OpenStack. It is OpenStack, Martin mentioned it. And there are just over 2,000 cores in the bare cloud facility. And it's something like 9 terabytes of RAM across all of the systems. And then we've got 3 petabytes of storage to underpin it as well. So why are we interested in doing cloud? So Nick was talking about, sort of gradware, software, things that are difficult. So we find as a research computing team, people come along and say, I need this application on the HPC facility that we've got. And we find it very difficult at times to get some of those applications working in there. So we're interested in how can cloud help us, it works fine on your Ubuntu system. So can we, use the cloud facilities to support additional research workloads? There's a lot of portals coming out. So we've heard Galaxy. Susan this morning was talking about portal access to facilities in the national e-infrastructure. So we kind of view that as being part of the sort of way of moving towards cloud-enabled researchers. So those portals, the code that you want to use, that you work with your researchers, how can we bring those into the facilities that we provide? The university also gave us a strategic investment. So they gave us a £2 million investment to help support Bear Cloud. So the life sciences people are one of these urgent drivers for why we need to do, cloud technology. How can we support them better in their research? We're interested in supporting golden science, so science reproducibility. Martin was talking about this. How can you take, the work that you do? Can we provide systems where you can take that and you can share that with other researchers? How can they take it away and run it in their institutions? Good research citizenship. So we see code that people bring along. It's not maintained very well. How can we use cloud technologies to help you build your systems in better ways, to help you support your code better, so that we get a longer-term research benefit right across the world, really. So some of the applications we get aren't really suited to high-performance, like MPI type works. So they're the kind of things that we run on Blue Bear. So again, cloud might fit in with that. People come along and they say, oh, it works fine on my desktop. Why does it take you 3 weeks to install the software? How can we ease people's transition from it running on your desktop now to you being able to run it in a higher performance environment or a higher capacity environment? So we view cloud as being a very good way of helping us achieve that. So what's Bear Cloud not? This is quite important. It's not an HPC facility. So we have Blue Bear. It's available now. But Bear Cloud is not a replacement just for that sort of MPI big processing workloads. Although we are keen, so if you've got research, there are research groups who need to run specific workloads in their environments. We're keen to talk to you about how we can help facilitate infrastructure as a service to support your research cluster. So rather than it sitting in the lab in the back end of a corridor, not looked after, not maintained, not supported properly, can we run that in our cloud environment and help you get a longer term benefit? Yeah, it's not a way of getting, just because the IT guys say you can't have admin rights on the PC, that's not really what we're intending Bear Cloud for. Well, the Nick says it is. And it's not like Amazon Cloud or something like that. It's not just for running web servers. Although, things like Galaxy, they're web portals. We're interested in running services and community services which, have computational or large memory requirements to help you deliver research and science. So our systems, they've got up to 20 cores in a single compute system. So those are real cores, that's not threads. We've got 128 gig of RAM in the current system. So in theory, you can request up to a complete node in there. So we run a research data network that spans out across campus. So it goes out to areas like the sequencing platforms, so that we're interested in how can they get their data faster across campus into the research data stores. So it's a fully integrated platform. It integrates with research data and so our data services. I'm ignoring Ed with his little red card. So our system's also got Mellanox InfiniBand in there. So we're able to provide InfiniBand low latency interconnects into those virtual environments that we're running. So I was asked to think, why don't we push people to use public cloud necessarily? So there are problems with doing that. How do we move a petabyte of data in and out of something like Amazon Cloud? There are costs associated with doing that. There's a whole bunch of IP questions, and so that's one of the things that the Working Group on Cloud, the RC UK funded, supported group, they're looking at, some of these IP questions, can we resolve some of those with universities and research organizations? But at the moment, we don't necessarily have clear answers to that. Capital expenditure can be difficult when it's on a service. How do we manage? So a researcher comes along and says, I want to have 20,000 hours, and at the end of the month, we get a bill from Amazon or Google Cloud. How do we manage that? So a private cloud, on-premise cloud, allows us to use capital expenditure to support that. A lot of the public cloud providers work on over-allocating memory and CPU. That's how their business models work to make it work out. So Nick didn't mention Climb. That doesn't over-allocate CPU or memory. We don't expect Bear Cloud to over-allocate CPU or memory. And other technologies, so InfiniBand, RDMA, MPI, GPU applications, they're not particularly well supported in public cloud providers. So we're interested in looking, so we don't have solutions for all of these yet. We're interested in looking at how we can support that when we own the whole architecture design. And generally, they weren't optimized. They weren't designed and built to run research workloads. So yes, research workloads were running some of the public clouds, but it was more of a byproduct rather than actually a specific going out and designing the facility to do that. So there are overheads from running virtual machines. We'll acknowledge that. But we've taken steps to try and work around some of those and to improve performance. So the Mellanox ConnectX 4 card. So there's a CAD diagram in the corner. So we heard Matt this morning from Lenovo talking about it, about co-design. We went to Lenovo and said, we want to use this card in the water-cooled platform. And we have the card. There's one on display upstairs. There's hardware offload functions in there. So some of the network functions that you typically, on a traditional cloud system, would see running on CPU, we can do that on the network hardware. So it frees up the CPU cycles for compute work. We have some very fast storage in there. So we've got guys from DDN who are supporting us today with the event. We have really fast storage under there, and there's a lot of flash in there to help alleviate some of the bottlenecks. So moving data in and out of VMs or running the infrastructure on top of the storage, we're doing some good work with them to try and improve some of the performance bottlenecks. And also, so this is a picture of inside one of the water-cooled chassis, if you take one apart. So this is the manifold where the water comes in at the back. There's a model on display. So we are the first UK site to deploy this technology, so do go and have a look at it. It's quite interesting. But because we use water cooling for energy efficiency reasons, we find the processors turbo more. So we've got a 2.4 gigahertz processor, so we tend to see around a sustained 2.9 gigahertz out of it because we're cooling it efficiently. And also changes in the Intel Broadwell platform, which are the latest generation of Xeon chips. So per clock scaling in there. So the impact across a whole core, across socket isn't the same. So where does Bare Cloud fit in the sort of bare world? Well, we expect to see user portals coming on top of there. So Jupyter Hub is you can do sort of Python code in there if you've not seen it before. You can have snippets and you can share those with other people. Galaxy portals, how can we provision Galaxy on demand? So we have a number of people who want to use it. So my colleague Lawrence isn't here today. He hates it because data security is awful. So running it in sort of silo-as cloud environments are good. We're hoping to bring GitLab along as well. So that's a version control system. So that's to help support you with your code. So we need to run it for our infrastructure purposes. We're hoping to expose that to researchers as well to help you build your platforms. There's a web portal. So I want to create a VM. I want it to be Ubuntu, and it's got to have 64 gig of RAM. So there's a self-service portal for doing that. I mentioned research group HPC instances inside the cloud. And it's connected across our Brocade research data network. So that spans out into sequencing platforms and other areas where you've got data, instruments generating high volumes of data. And then we've got Bluebear HPC. And we're interested in looking at, if there's spare capacity in the cloud, can we use that for running our Bluebear HPC for certain types of workload? And we think we can probably do some of those things. So we can use the capacity to support general HPC work as well. And we have our research data platforms as well. So Martin was talking about research data services. We have a large storage facility available, and we have archive facilities available as well. So where do we want to go next with Bare Cloud? So if you've got research portals, code that you want to use, other things other than Galaxy, come and talk to us about, those workloads, those portals that you want us to help develop. We're looking for doing community support type activity. So we'll provide some level of support, and we look to research groups to provide us assistance as well. So containers, Docker, Kubernetes. Can we provide support for Docker? So Docker it's got some interesting security requirements. Can we provide a more secure Docker environment? And I think, so we've got a speaker this afternoon, Dean from IBM. So Dean's in IBM Research. He did a lot of work with us when we were doing Climb, on getting Climb up and running. So he's going to talk a little bit about containers. How do we help science reproducibility? How do we make sure that you can move your VMs, platforms across different systems? And can we do GPU enablement? We think we've got some ideas about how we can do that. But is it something people want to see in the cloud environment? Do you need GPUs in the cloud? And then in the future, how do we make it work with other research clouds? So, can we make some of the portals disappear so that they, work goes onto climate when it needs to, or it goes onto other research portals, or even public cloud in the future? And I will shut up now, and Ed can put his red card up again.